The lecture you are about to see is part of our annual Alan Edwards Psychology Lecture Series. Professor Edwards was affiliated with the Department of Psychology for half a century until his death in 1994. He was an outstanding teacher, researcher, and writer who introduced new statistical techniques that are credited with changing the way modern psychological research is conducted. Alan also permanently enhanced the intellectual climate at UW Psychology by endowing the Alan Edwards Lectureship, which since 1999 has brought in an impressive list of renowned psychologists to the UW campus to interact with faculty and students. The lecture you're about to watch is one of a pair given back-to-back that matched a UW psychology professor with a visiting researcher to talk about a topic of great public and scientific interest.
Please join me in welcoming Dr. Sheri Mizumori.
Thank you. If I were to ask you to think of a memory, a variety of experiences from across your life might enter into your mind. Sometimes these memories would be so vivid that when you recall them, you would feel like you were re-experiencing the event. Other memories may feel vague. That is, it seems like there might be many details that are missing. For all events in your life, however, you can't help but form some kind of memory about it. All of these memories combine to define who you are today. They are, in fact, the essence of our self-identity. Imagine for a second what it would be like if you did not have your memories. How do we study such an abstract and yet very real construct such as memory? Well, in order to answer this question, we first need to ask the basic question of what are memories? Well, according to Webster, memories are stored information that is later recalled. Okay. Well, this definition does not tell us how our memory systems work, and they do not reveal the richness of this phenomenon. For example, we often wonder how are memories formed? We ask, do they change over time? And if so, how? Are memories really forgotten, or are they just deemed inaccessible for periods of time? Can they become momentarily accessible later on? And is there a limited capacity to memories? Well, these are fundamental questions that psychologists ask so that they can better understand how our past experiences guide our current behaviors and our choices in life. Neuroscientists ask similar questions as they seek to understand how the brain accomplishes these remarkable and essential functions. Together, both psychologists and neuroscientists ask, what are the consequences when brain systems that mediate memories fail? That is, both scientists are continuing to seek ways to treat conditions that are associated with poor or at least less than optimal memories, such as those that result from brain trauma or injury, or that occur with just normal aging or pathological states. And a clear understanding of how memory works is also important for policy making in our society. For example, the determination of admissible evidence in court may depend in part on our belief that accurate information can indeed be recalled under stress. Now, to answer these frequently asked questions about memory function, either from a psychological or a neuroscience type of perspective, it's useful to think of memory in terms of its components. Now, the image that you just saw flashed on the screen entered into a very short-term memory that you have, which is called a sensory memory. This lasts for maybe 2 or 3 seconds. But this memory can't help but fade away. And as you continue to listen to me, you can probably feel the memory fading away again. Now, attending to this information that appears in your sensory memory here allows it to enter into a little bit longer lasting store referred to as short-term or working memory. Then in working memory and short-term memory, you're able to hold on to information for maybe about 10 or 15 minutes. And you can retain this information in the short-term memory by a process called rehearsal. And some of the information held in short-term memory eventually makes it into a long-term memory store via a process called encoding, sometimes also referred to as just the transfer of information. Now, how information is selected from the short-term memory to be put into long-term memory is not completely clear. But we do know that information from long-term memory can be brought back into a short-term memory region so that you can think about it, add information to it before it goes back into long-term store again. Okay. Now, what happens if you view a variation of a scene that was familiar, but it's not exactly one that you've seen before? And let's say you were asked to identify what it was. Well, it might take you a little bit longer than seeing a familiar scene. And the reason is as follows, is that initially the information comes into your sensory memory, eventually into short-term memory, and then some of that information will go into long-term memory, okay, in order to find the name. But if there is no name there, there is no match between the scene and the names that you have in your memory. And as a result, you will pay closer attention, perhaps, to the scene to try and abstract out more details. And as you do, more information comes in, you're able to fill in more of the details in the short-term memory, and eventually, if that information leads into the long-term stores where the long-term memories become updated. Now, at this point, because you really haven't seen this picture before, that what you'll have to do is make a decision about a name based on uncertain information. Okay? And so at some point, after you've acquired a little bit more information, you'll take your best guess. Now, for those of you who are familiar with the university campus, you will know that this is Red Square, but you may not have seen Red Square with snow on it. Okay? Now, what happens when these essential processes go awry? Well, without good retrieval mechanisms here, you're not able to pull the long-term information into a short-term memory. And as a result, you can remember information for a short period of time, but you're not going to be able to recall. It's possible that short-term memory will have a problem being encoded into long-term memory, okay? That this encoding mechanism here might go awry. And as a result, again, you can only remember information for a short period of time, and it will seem like information then fades away after about 10 minutes. It's also the case that maybe information does go from short-term memory, gets into long-term memory, but somehow has a hard time getting embedded in that long-term memory structure. So once again, you're going to feel like, I have the short-term memory, but I can't seem to remember things for very long. So this is an illustration that a failure to remember information could be caused by a number of different problems. And even today, the scientists debate whether a particular case of memory failure is due to problems with memory encoding, with updating, or with retrieval. And often, it's really not very easy to determine which one it is. Now, a common question that I'm often asked has to do with whether or not you can improve your memory. And as you know, there are numerous attempts out there to convince you that you can. But consistent and strong scientific evidence is sometimes hard to find. And for that reason, I was quite excited by a paper that came out that suggested that maybe that there is a different strategy that one can take to try to improve memory. Now, this came out in a paper that was published in the Proceedings of the National Academy of Sciences, a very prestigious scientific journal. And this was described as a study by the Junitas Research Group at the University of Michigan. Okay? And what they did was they suggested that if you were to somehow practice these processes of retrieval and rehearsal, that you can actually improve your ability to recall information from your long-term memory stores. Now, you might say to yourself, I think we already knew this. We already knew that if you continually recall stored information, that you would strengthen a memory. In fact, this is what we do when we purposely try to remember something, right? We recall it over and over again. Well, let me describe the experiment to you, and then I'll tell you what was a little bit different about it and why it was so remarkable. Okay. This is the question that the subjects were asked. They said that, well, we're going to be shown this series of objects on a screen. And you are to indicate when an object that is shown is the same as the two objects before. Okay, well, this was the series that I just showed you. And the correct answers would have been this ball, the second ball shown, because two objects before was a ball. The next correct answer would have been this picture of a flower, because two objects before was a flower. Okay. The individuals participating in this experiment then performed many of these such repetition types of trials. Well, it ends up that if you were to perform this task over many different training sessions, okay, and we evaluated the accuracy of your performance as shown on the y-axis of this graph. Some of you may have fallen in this group, which is shown by these open circles. That is, some of you may have improved over time with training. But notice that some of you may not have improved, and they may show a relatively flat curve with training. Okay? So what this tells us is that not everybody will benefit from the same kind of memory training. So that's a point to remember. Now, the researchers from Michigan were interested in whether this kind of repetition training impacted future memory use in a different way. They were interested in whether or not this kind of repetition training helped you with problem-solving ability or the flexible uses of memory. And so they divided these subjects into those that benefited from the repetition training and those that didn't. And they looked to see what their performance was on a flexible memory task. And high scores on this graph indicates that they did quite well. And you can see a huge difference. Those individuals that benefited from this repetition training showed greater flexible memory use than individuals that showed less benefit from the repetition training. Now, these data were collected right after the repetition training sessions. If you wait 3 months, the interesting thing is that you see that this difference persisted. That is, those individuals that benefited from the repetition training 3 months before still showed a savings and still showed flexible memory use that was higher than the individuals that showed less benefit from this training. Now, this is quite interesting because this effect can be likened to the physical domain of exercise. That is, one can run to increase the cardiovascular tone. And the positive effects that you see can be seen not only in your running ability, but in other physical activities such as swimming or biking. So there are a couple of take-home messages from this experiment that I'd like you to think about. And one of them has to do with the fact that not everyone will benefit, again, from a particular method of improving memory. And this likely reflects the fact that each of our memory systems has different patterns of strengths and weaknesses. Also, from this Janitas study, it shows that maybe if exercising certain processes of memory, like rehearsal and retrieval, and perhaps using the special repetition procedure, that this can have a general positive impact on the person's flexible use of memories in the future, and not just memory for the particular items that were used in the repetition task. Now, this result is fairly new, and of course, still it needs to be replicated and evaluated further, but the idea is quite intriguing. Now, as a behavioral neuroscientist, I'm interested in how this homogeneous-looking structure here, the brain, helps us and endows us with the complicated skills that we know memory systems have. Our first insight into this issue comes from the study of brain-damaged patients who have memory disorders. And one particular patient is worth discussing in more detail at this point. The most famous amnesic patient who recently passed away after spending over 50 years revolutionizing the way that we think about how the brain organizes memory, we used to call him, when I was in graduate school, just patient HM. Once he passed away, we learned that HM was really Henry Molaison. Here he is as a young child, around 1930, and about that time, it was noticed that he experienced in his brain abnormal, irregular, and uncontrollable high levels of neural activity, shown here as epilepsy. And this epilepsy just continued to get worse and worse with time. He did finish high school, but later than normal. And he could not hold down a job as an assembly worker, and he could not maintain friendships. He experienced major epileptic episodes that include huge convulsions and lapses of consciousness that lasted for many minutes. His condition seemed hopeless because he was not responsive to drug treatments either. Thus, he underwent what was then a very radical and experimental surgery to get rid of the part of the brain that was generating the seizures. That is, the neurosurgeons went in and removed a structure referred to as the hippocampus. The surgery worked, okay, but in that the epileptic seizures were much more under control. However, he paid a terrible price. Okay. Now, before I talk to you about what some of the memory problems of HM were, let me just show you that in the slide down here near the bottom, you can see that there are these holes in the brain, and this represents the areas of the brain that were removed by the surgeon in order to control the seizures. Now, if you were to take an image of the brain and cut it in half, so let's say you cut it in this plane here, let's say you were able to open up your head, look at it from the inside out, then you would see a section that looked like this. And these holes down here represents the gaps where the hippocampus used to be. And this compares to the intact brain shown on the right. Now, after the surgery, as I said, the epilepsy was more or less controlled, and patient HM was able to continue to live in Connecticut for most of his life. But then in the 1990s, he was placed in a retirement home, and he unfortunately passed away in 2008. Now, immediately after the surgery, it became very clear that HM had serious memory problems. He could not remember anything that happened to him, any event that happened in his life after surgery. Okay? And this persisted until he passed away. He could carry on a normal conversation. He appeared to have normal intelligence. He had appropriate social skills in carrying on, in that he was very pleasant to talk to. But after you talk to him for about 10 or 15 minutes, it would seem like he was waking up from a dream, and this is how he described it. And then because the memory that he had just happened to fade away forever. Even after decades of meeting the same doctors, he did not recognize them or their names. He could not tell you what he had for lunch, and he could not talk to you about current events. And although HM suffered memory loss for some events that happened prior to the surgery, this type of amnesia was relatively short. That is, if you look further enough back in time, you find that HM indeed remembered his childhood and some of his long-term memories. Now, the fact that HM suffered memory loss was quite striking. But the topic that really fascinated scientists for decades after HM's surgery was the fact that his memory loss was selective. That is, that it became evident that there were certain things that HM can learn. Now, this was revolutionary because back in the 50s, researchers thought that there was only one memory system and that there was only one part of the brain that contained this memory system. So HM told us that this was just simply not the case. So let me give you some examples of what it was that he could do. This is referred to as a mirror drawing task, where the task is to take a pencil and to draw a line in between the parallel lines that outline the star. Simple, you say. Well, what if you couldn't see the paper that you were writing on, and there is this barrier here preventing you from doing so? But you can look at the image, but look at it in its reverse image through a mirror. Okay, not so easy. So what you would find is that if you started to do this, your lines would go outside of these parallel lines, and those would be considered errors. And in the beginning, you might make lots of errors until you acquired the appropriate motor skills. And after a few sessions, typical individuals would get better, and then they have it down, just like riding a bicycle. They would be able to do these mirror reverse drawings, no problem. Well, it ends up that HM could do this as well. Initially, he made lots of mistakes, but in about the same amount of time, after a couple of sessions, he improved, and then he stayed quite good at this. Well, the interesting thing is he never remembered coming into any of these sessions. So at the end, when you ask them, how is it that you can do so well if this is the first time that you've done this? And he would say, I guess I'm just naturally a good artist. Okay. This is just another example of the fact that information did get into his memory system. This is a transcript of a conversation that HM had with Brenda Milner, who's a well-known neuropsychologist. Brenda Milner had worked with HM for many, many decades trying to evaluate his memory abilities. Here's Brenda saying, someone was assassinated in 1963. HM says, well, he'd been a president. Milner says, that's right, what was his name? HM, he had been, like you said, he had been a president. Milner says, well, his initials are JFK. HM says, Kennedy. That's right, what's his name? Milner says. HM says, John. So clearly, after his surgery, he was able to learn this bit of information that John Kennedy was a president and that he was assassinated. Okay? Another example of his capabilities was the fact that he was able to recall those memories from long ago. And so his retrieval processes must have been intact. They don't require a hippocampus, in other words. He also knew that he had a memory problem. In fact, if you couldn't answer a question, oftentimes he would say, well, you know, I have a memory problem, so I can't answer that question. So he clearly learned and remembered his condition. Okay. So to summarize, HM's deficit was quite selective, and he had difficulty remembering events that happened to him. Nowadays, we refer to this event-based memory system as episodic memories. But he was able to remember other kinds of information. He had intact knowledge or semantic memory, and he was able to learn simple sensory motor skills. That is, he was able to develop sensory motor habits and remember it in what's called a procedural memory. So I want to mention a couple of lessons that we learned from HM. and that is that there are multiple forms of memory in the different brain areas, and also that hippocampus and also its surrounding tissue, referred to as the cortex, is essential in order to learn and remember events and to develop new episodic memories. Okay? Well, how does the brain accomplish such a complex processing, such as the complicated computations that must come into play when you have event memories or developing episodic memories? Well, with the new technologies now, we've gained some startling new insights that could change the way that we view memory processing in the brain. Okay. Well, nowadays, for epileptic patients, the neurosurgeons do not remove the whole hippocampus, but rather they go into the hippocampus, as shown here in red by the side view of the human brain and also shown from the top view of the human brain. So you can see you have two, one in each hemisphere. And what the neurosurgeons do is they place a fine electrode into the hippocampus structure shown here, okay, by these particular shapes. Okay. And what they're doing when they do this is they're placing an electrode to listen to the electrical messages that neurons, the cells of the brain, send to one another. In other words, it's a way of eavesdropping to see who is screaming out way too loud, hence the site of the epileptic focus, or who's carrying on normal conversation. And when they find the sites where the neural activity is just uncontrolled, then they know that that's the area that they need to get rid of. And as a result, for treating epilepsy nowadays, they do go in and remove hippocampus, but only parts of hippocampus. And so we don't see the same kind of severe memory problems that we saw with HM. Okay. Now, it's interesting. This is an example of Dr. Itzhak Fried, who's a neurosurgeon at UCLA. And this is taken from a NOVA show that's in 2009. So if you have a chance to look at it, it's quite interesting. But I wanted to show you an example of a patient who was hooked up to these electrodes so that Dr. Fried could find the epileptic focus. Well, what you find is that this procedure of looking for the focus is harmless and painless. Okay? Numerous subjects give permission to participate in memory experiments while this exploration procedure is going on. Now, if you were in the room when this was happening, you might hear something that sounds like this. That is the sound of neurons sending messages to their neighbors. They're electrical discharges. Let's hear it again. Pretty remarkable, isn't it? I tell you, the first time I heard it, I was just hooked. I just had to figure out what these signals were. Now, in one of Dr. Fried's experiments, a subject was asked, and so this is the same subject that you saw on the previous slide. A subject was asked to identify and look at these different scenes from well-known TV shows or movies. And at the same time, these hippocampal neurons were recorded. Let's say that they were listening to one cell. The cell might respond to the scene from Jurassic Park in this way. Pop, 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 pop. Okay? And then The Simpsons would come on. The cell would respond by saying, broot, broot. Very excited. Well, The Wizards of Waverly Place comes on. And they say, and the cell just goes back to its low rate, bup, bup, bup. Star Trek comes on. Nothing. Bup, bup, bup. Clearly, this cell preferentially fired, is how we say it, to this particular scene for some reason. And it ended up that when Dr. Fried records from lots of cells at once, that there's always a subset of cells that seem to prefer this particular scene. Other subsets of cells might prefer Jurassic Park, okay, or Star Trek. Well, this is remarkable, okay? But what was even more surprising is what happened next. What Dr. Fried did was he took the same patient, listening to the same cells, put away the computer, and said, okay, now think about those scenes that you just saw. So the patient would think about the Jurassic Park, and what you would hear, because you're still listening to the brain, would be this pattern of activity. When she thought about The Simpsons, brrrt, brrrt, the cells would fire like crazy. When she thought about these other two shows, again, a low rate of firing. So what this says is that these cells were not responding to the visual input of the scene, okay, because you can just think about it without having the scene in front of you and the cells would still fire. Okay. And so Dr. Fried concluded that memory is represented not only by this individual cell, but but it's because we saw a lot of cells responding to this particular scene that memory must be represented as a coordinated network of connected cells, and that during recall, this network can be recalled and replayed, okay, as you saw it in its original form. That's pretty striking. Okay. Now, this next slide illustrates a cartoon, okay, that talks about how these memory networks might work. Now, initially, just imagine that on the left here, that each one of these little diamonds are individual neurons. Now, let's say that the individual starts to learn some new information, and there's some subset of cells that will start to become active together. They'll start to form connections and start to form a network. As the memory becomes stronger, the connections between these cells will get stronger and stronger. In fact, they get so strong that if you were able to come in and activate just one of these cells, the entire network would be reactivated. It would be recalled. Okay? Now, what would happen with one scene, as shown in the top row. What if you were viewing another scene, as that patient that I just described? Well, then it might be then a different set of cells might become active, as shown in the middle down here. It would become active during initial learning, but the connections between that set of neurons would also become active. So again, showing that when the memory gets stronger, then the connections are probably stronger as well. Now, you may also notice that since these are the same, in the top row and the bottom row, that these are the same cells, that there's a neuron that's shown here on the right side of this blue sort of triangle, and this one here on the left, that this cell is participating in more than one network. Okay. That does happen. And this may actually have great benefits, because this may allow us to develop relationships between different memories via the shared elements and components of a memory representation. In this figure, what I was trying to illustrate was the fact that networks of neurons can develop connections with learning, and that these connections become stronger as memories become stronger. And that the pattern of neurons that become activated can be different for different memories. So across the top row, this illustrates one type of memory network that might be active, say when that patient was viewing a scene. And on the bottom row, this represents another memory network that might be activated when the patient was viewing a different scene. And what was interesting was that sometimes there's an overlap in which cells would contribute to one network and another. Okay, in this case, that would be a cell here on the right and this cell on the left of this particular assembly. And this ends up having possibly great benefits, because this gives us some insight into how the brain develops relationships between different memories just via these shared elements of a memory representation. Now, a question is this replay of neural activity that I just described, is this found in normal brains, that is non-epileptic brains as well? Well, we don't know the answer to that question yet, because for humans, we can only probe human brains when their brains are exposed for medical reasons. And because of the way that human subjects are tested, it's difficult to assess whether memory replay in the brain contributes to other dynamic features of memory function. Okay. And thus, scientists have taken advantage of the fact that the hippocampus is what's called an evolutionarily old structure. That is, it's present in all vertebrate animals. And not only that, it looks very similar, and it seems to have similar functions. So in the humans, as I said, as we saw before, in red, this represents where hippocampus is located in humans from the side view and then the top view. In the rodent, from the top view, you can see that here's the hippocampus, and from the side view, this is also the hippocampus. Okay? It looks a little bit different in size, but if you were to look at the structures of the hippocampus itself and the organization of the cells within it, you see there's actual remarkable similarity. These dark lines here are areas of cells where they're all clumped together in this line. And there's another one here. It's as if there's two interdigitated C's. If you look in the rodent hippocampus, you also see that this is one part of the C and this is another part of the C here. Okay? There's again, interdigitated organization of cells. And so given that these structures are very similar and they seem to have similar functions in that rats also need the hippocampus to establish event-based memories, then scientists can use a very similar method in rodents and humans to try to understand how memories might be encoded in the brain and understand the function of replay. So in rodents, one can also put in an electrode and listen to what the cells are saying to each other. Now, we can't not ask rats to watch TV, like The Simpsons, or to watch Jurassic Park. But we can teach them to make visual-based associations. That is, and rats are actually quite good at this. They can learn to associate particular scenes with the location of something very important to them, chocolate milk. So in laboratory tasks, you might have rats that will run a particular maze. This is referred to as a plus maze. They might start, for example, on the north side, the west side, the south side, and then they just need to come to the center and go out here to the end of the east arm to find food. Now, the question is, if an animal is performing this kind of visual association task, what are hippocampal cells doing? Okay. Well, it ends up that when you're recording a hippocampal neuron from a rat that's performing this task, it shows a pattern of firing that looks like this. In this plot, this little crisscross that's shown in the blue represents the area of the maze. The locations that are associated with high firing rates of the cell are indicated by this red and yellow region here. And so what this shows is that when the animal is on the north arms, the west arm, or the south arm of this maze, the cell doesn't fire much at all. It's kind of like that cell in that patient and when the patient was watching Star Trek or Jurassic Park. Okay. However, when the animal moved down this east arm, what you find was that the cell went crazy. Okay. It exhibited what's referred to as location-selective firing, and the location that caused the cell to fire is called the place field. The cell that is actually firing is called a place cell. So place cells have place fields. Now it's interesting that if you look at times when the animal started from the north arm only and then plotted the place field, or whether or not there was a place field, and compared that to other times when the animal started from the south, you saw a really interesting pattern. Depending on the start location determined whether or not you saw a place field on this east arm. So what that means is that this particular cell was taking into consideration the memory of past behaviors of the animal before it decided to exhibit a place field. It can be memory driven, just like what we saw in humans. The fact that we can evaluate place fields as a marker of the kind of memory representations in rodents has led to some very startling discoveries, and it has allowed us to study this replay phenomena. So here I'm going to describe to you an experiment that showed you how we can study this replay. So just imagine what we call just a pre-learning condition. So in this case, this mouse is asleep, and let's say that we were able to record the hippocampal neural activity. Imagine that these little circles up here near the top of the slide are individual cells, and that these cells are connected in sort of random ways, and when the mouse is not doing anything, the cells are firing off in random ways. Okay? If you could plot the firing of these cells as a function of time on this middle plot, you would see that the different colored cells would appear at sort of different times, again, random relative to one another. Now, if in a subsequent learning session, you taught the rat to do a very simple task. In this case, what the rat had to do was to go down this particular alley to get to food at the end of the alley. Okay? Now, as the animal is moving down this alley, it's going to go through a place field, a number of place fields of cells in this network. And it will go through them in a particular order that makes sense in terms of going from one end of the arm to another. So when he goes into the red place field, the red cell will fire. When he goes into the blue place field, the blue cell will fire, and so on down the road. Now, during this test, the animal goes down this track many, many times. And if you were to watch the relative firing of the cells, you would see that, oh, the red cell fires first, followed by the blue, followed by the green, and the orange, and the purple. The next time the animal goes through, the same sequence is fired, okay? Maybe that's not so striking. You say, oh, well, that just means that you're organizing, the cells are responding in a way that corresponds to the behavior, okay? But what was really surprising is that in the next phase of the experiment, the animal was asleep, and now we record the cells, the same cells that were recorded in all these previous two sessions. And what you find is now there is a temporal order, a particular sequence that these cells are replayed in during sleep. And this replay, this order matches the experience of the animal before they went to sleep. It's no longer random. So there's a number of implications of this, and that is, one implication is that scientists have interpreted this sequential organization during sleep as an example of rehearsal of previously learned information. And of course, this has implications for ideas that sleep facilitates learning. And it might facilitate learning because it allows you to rehearse particular memories. And this is an example of a rehearsal because we're replaying the neural activity patterns that were shown during the learning itself. Again, I think this is amazing that these sequences were found. Okay? So sequences are very, very cool. And now we know a little bit more about this. That in the rodent hippocampus, we find that there are what's called forward sequences. That is, let's say an animal is still going down this track, and because he has these sequences in mind, wherever the animal is, then the memory just needs to be called up, and it can tell the rat, it can predict to the rat what the next place should be. It can end the next place and how many more places it needs to go to before it gets to food. That's called a forward sequence. It's telling the animal where it needs to go. When the animal gets to the food, we also now know that there are what's called backward sequences. That is, from a particular place in space and time, if you were to look at the same cells, record the same cells, they will replay but in reverse order. So now when the animal's at the end of the arm, the purple cell will fire first before the orange, before the green, before the blue, and before the red. But it will go in that particular sequence. Again, just remarkable. And what does this mean? Well, scientists have suggested that this may be a mechanism by which one can predict the future. Okay, and that's a function that's really important for memories. And not only that, but it can allow you to look back on the past, okay, and allow you to call up sequentially your past memories. Okay, so up until now, I've described hippocampus as representing memories in terms of networks of cell activity, and I've described how hippocampus can encode sequences of information, okay, that reflect and are driven by your memories. Okay. Well, you might be asking yourself, how do these kinds of representations relate to the problems that HM had? Okay. Well, it ends up that these sequences can be used to detect changes in a given situation or a given context, and that it may be critical for episodic memories to be able to understand when one event or one context ends and the next one begins. Let me just give you an illustration. So here again is our rat who's running down the maze and engaging in this sequential kind of activation of cells. And its memory is telling it where to go next. And when the rat moves into the next space, let's say into this place of the orange field, the memory is updated because now the next space is this purple spot. Okay. Now you're using the same memory to keep driving these behaviors. And so the hippocampal output would say, oh, okay, this is a match. It's good that we're still using this memory. Let's maintain this behavior and strengthen the current memory networks because it's working. Okay? But what if the animal gets to the end of the arm and something's different? Now, instead of food, there's an extra set of options available to it. This is something very unexpected. So when the animal is here in this last spot, its memory is telling it, oh, there should be food, but oh no, it's not. It's not there. Okay? So the hippocampus detects this unexpected event as a mismatch. That is, what happened does not match what the memories were expecting. Okay? And as a result, a different message will go out to the rest of the brain so that it can start the process of evaluating the significance of this change. So without a hippocampus then, you could not make accurate predictions about when one event will end, and then, of course, when the next event begins. And if your world seems unpredictable, how would you react? Okay, this dilemma may have importantly contributed to the impaired episodic memory function of patient HM. Now, I want to talk about more this comparison function of the hippocampus. And one way to think about it is in this following little cartoon that I'm going to now draw. Let's first ask the question, well, hippocampus is detecting changes in your environment, detecting what's referred to as context changes. And so hippocampus can answer this question. It can answer it as, no, there hasn't been a change, continue on with the same memory networks. Or it can answer, yes, there is a change, and now we need to assess the value of this change. And it does so by activating a whole other set of brain structures that we think is involved in what's called decision making. That is, allowing you to make a decision about whether or not this change in value is important enough to cause a change in behavior. So this next brain area answers that I'm going to talk about answers this question about or detects the value of this change. Okay. Now, in principle, if this next area that I'll describe in a minute comes up with an answer that says, well, despite this context change, the reward was still there. So no, nothing changed, so we don't need to change our memories. We still are getting the reward that we thought we would get. On the other hand, if the value has changed, then it needs to update the memories accordingly, so that the next time that the animal or you would encounter the situation, you'll be able to respond with the most recent updates. Now, it ends up that there's a structure in the brain referred to as the ventral tegmental area. Okay, again, you don't have to worry about the name, just ventral tegmental area, and we're interested in particular in the dopamine neurons that reside within this ventral tegmental area. And this is just a picture of one of these dopamine neurons. Because it ends up that the firing patterns and the signaling of these dopamine neurons ends up being very critical for your assessment of the value of experiences. Now, in a case where, let's say you're in a particular environment, nothing much is happening, you're just kind of sitting there, if you were to record these dopamine neurons, they would fire maybe in a pattern like this. They would just kind of go bup, 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 bup. This is kind of a low rate, random firing pattern. But let's say now you take your rat and put them into this new environment. Okay? Oh, this is different. Okay? Hippocampus notices something's changed. So it will send a message out to this value assessment system that says, okay, something's different, listen up. And as a result, the same cell that used to fire at this low firing rate now starts to fire when the animal encounters rewards that it might find at the end of the maze arms. Not only that, the responses that it sees here is proportional to the amount of the reward that the animal gets. So if the animal goes down certain arms and gets a lot of reward, you get a huge response of these dopamine neurons. If there's a little bit of reward, as indicated by these little dots over here on the maze, then you might get a little bit of reward. But this combined pattern tells scientists that these dopamine neurons are actually encoding reward values. Now, as I said, when hippocampus is activated when you go into a new environment, it says, aha, here's alert, pay attention. But as you learn this task, it becomes familiar and things don't change anymore. Events happen as expected. And then as a result, the hippocampal message will be, you don't need to change anything, the memory's working. And as a result, the message comes down to these dopamine neurons, and indeed, this same cell that used to fire to rewards no longer fires to rewards. Now, let's say, because sometimes unexpected things happen, let's say that one of these rewards, instead of containing 1 drop of chocolate milk, contained 20 drops of chocolate milk, a real gold find for these rats. Then what happens is, well, the dopamine cell must have been paying attention to the reward values, even though it wasn't really saying much in this familiar condition, because when the animal encounters this unexpected reward, the dopamine neurons go crazy. And you see this very large response. But again, the response is graded. Okay? When it encounters a very, very large one, you get a very, very large response. When it encounters a medium-large sort of new reward, it then fires at a little bit lower rate. So again, we consider this an example of reward encoding by these dopamine neurons. Now, sometimes you go someplace and you expect a reward and it's not there. And that can be just as effective in changing your behavior as when you find a reward, right? Well, these dopamine neurons encode that as well. So the same neuron that was encoding rewards with this excitatory response will now go quiet. They'll become inhibited when there's a negative reward. And the duration of this inhibition is related to the degree of sort of negative impact, you might say, of these cells. So again, it seems to be encoding reward value. But it does so in different ways, depending on the situation and depending on the context. Let me just give you an example. Let's say that you came into this room and you found $20 bills sitting on your seat. Your dopamine cells would go crazy, okay? Because this was highly unexpected, okay? Now, let's say that you come back many, many times, once a day. You keep coming back and you keep finding these $20 bills. Well, for the first few times, you're going to be pretty excited. But after about 100 times, you still might be excited, but it's going to be a little bit less than before. In fact, you're going to come to expect that this $20 bill is going to be sitting on your seat. And when that happens, your dopamine cells are not going to be firing so much anymore, okay? Because this is expected information. It may be valuable, but it's expected, okay? Well, let's say you came in the next time and there was $1,000 sitting on your chair. Your dopamine cells would go crazy again, right? Because it's unexpectedly high. On the other hand, if you came in and the $20 bill was gone, the cells would be silent. You'd be inhibited. Okay? So that's an example that is parallel to what these dopamine neurons were doing in the rats when we were running them on this particular maze. Okay? And again, the striking thing is that the same cell can show such a dynamic range of responses. Okay, so value coding by dopamine neurons. Then what happens then is that it engages a whole number of other structures. But in some, those structures have the job of determining whether or not what the consequences will be of this particular reward. Will there be short-term gratification? Will there be long-term benefits? Are there costs and risks associated with this particular choice? Okay. Now, this is a very complex procedure, as you might imagine. But there are some very insightful ideas that came from a couple of well-known scholars. In fact, Daniel Kahneman was a Nobel Prize winner in economics. And what he found, he and Amos Tversky described back in the 70s, was this idea that the brain tends to reduce the complexity of situations when there are lots of unknown factors, perhaps, in order to optimize decisions as quickly as possible. Okay. And what they suggested was that there are certain processes that the brain automatically engages in. One of them is that it automatically assesses similarities between this situation that you find yourself in and others. That is, it looks across the memories to see if they can maybe learn lessons from other situations. It also seems to automatically create patterns among seemingly random bits of information out there in an attempt to try to make sense of the cues, the new information that's out there. It's also the case that choices that one makes is strongly biased not just on whether or not that reward is there in front of you, but by the context in which it's framed. So for example, if someone came up to you and asked you for $10, well, if you had $1,000 in your pocket, you may not mind giving them $10. But if that $10 was your last $10 that you had, the chances are that you might not give them that $10, right? It's the same action, they're making the same request, but your decision is based on a different context. So for now, what I'd like to do is change gears a little bit and go back to this diagram of this flexible processing that we saw the dopamines engaged in. And this fact that a single cell can show these many different responses on such short notice, so to speak, is referred to as neuroplasticity. That is the flexible responding, the flexible behaviors, you might say, of neurons. Okay. And what we find is that neuroplasticity ends up being very important for learning. Again, maybe this is not too surprising. When you learn, changes happen in your brain, chemicals change, connections change, and so its neuroplasticity ends up being really important. And so it's not so surprising, probably, that there's been tremendous effort to try to understand the cellular and the molecular mechanisms of neuroplasticity. Moreover, now, and you maybe have heard some of this in the press, that there's a number of ideas about how your behaviors, for example, exercise, can alter the proteins and the chemicals that are involved in neuroplasticity. Okay. And in fact, now I think that there's pretty convincing data that it does. The question is whether it does it in the way that you want. Does it allow you to learn the kind of information that you want? But we know that it enhances, that exercise would enhance memory function. Okay. Now let me just illustrate in a cartoon fashion this plasticity that's related to learning. So here on the left, you see, in the upper left, you see again 3 cells, but this time what I've done is I've added processes on these cells, and this is important because it's these processes that are connecting one cell with another. Now with learning, a cell may send out additional processes to make connections that weren't there before. And as learning gets stronger and new long-term memories are made, these connections become more permanent. Now what happens in conditions such as aging, in what I call non-pathological, normal aging? Well, surprisingly, the number of cells in an aged brain doesn't go down nearly as much as what you might think. But what seems to happen is that a lot of these processes seem to go away. Well, these processes are the ones that are responsible for some of this plasticity, right? Up here, I'm just illustrating how these processes are what's growing and making connections. Well, it's interesting that in an older brain, when there's new learning happening, that the remaining cells may extend out additional processes as if to compensate for the loss of flexibility in these other cells that have lost processes. And then as those long-term memories are formed and created and gotten more stronger, you see that those connections become more permanent. And this is actually good news. This is good. So this means that even though there are age-related changes in the cellular physiology of the neurons in your brain, that the plasticity mechanisms are still intact, and this can allow you to come up with alternate ways to learn and remember information. I find that very good news. Now, what that means, though, is that if you're testing memory in older individuals, it may be more subtle than you think. Okay? This is one example of a recent publication that illustrates this point. So in this study, this was a study carried out by Veronique Bobo, a colleague in Montreal, Canada. And what she did was she developed a task to test spatial memory abilities in normal aged humans, but also comparing them to young, healthy individuals. And this task here was taken from that maze task that sort of starburst maze task that I showed you before. So this is a virtual reality test. The person is sitting in front of a screen, and their job is to move around this environment here. This is a top-down view, and they can be placed in the center. And the center view looking out shows these arms that are extending out. And just like in the rat studies, there are rewards placed at the ends of these arms. And so in this task, in one trial, the subject may be shown two arms, let's just say arm 1 and arm 2, as shown here. And what they learn is that when they're shown arm 1 and arm 2, arm 1 is always associated with the reward. Arm 2 is never associated with the reward. And so anytime they're shown this pair, they know to go to number 1. They are also shown the pairs of 3 and 4, where 3 is always rewarded, maybe 5 and 6, where 6 is always rewarded, and so on, until all the arms are used. Now, with time, it takes a little bit of time, but with time, individuals, young and old, will learn this, what we call paired associate learning. Now, when this task is learned, then comes the next stage, what's called a flexibility probe. And in this task, the subjects are shown a novel set of arms. So, for example, 2 and 3. This is not a combination that was shown before. But if the individual is remembering the outcome of going to each one of these arms, they should pick arm 3 because arm 3 is the one that was rewarded before. Okay? Arm 2 is not, and so that arm 3 is the correct choice. When they're shown 6 and 7, arm 6 is the one that was rewarded before, so that's the correct choice again, and so on down the line. Another way to test spatial memory in humans is to use a virtual navigation task. And this is just one example of the kind of scenes that the individual might be exposed to. So rather than looking at this maze, the individual may be allowed to search through a virtual town like this in search of rewards. And then there will be locations that are always associated with rewards and other ones that are not. It ends up that regardless of which way you test them, the result is the same. So this one was taken from this maze task. Now in this case, individuals were trained on these paired as a function across many different trials shown here on the x-axis. The percent correct is shown on the y-axis. If you look at these dark circled line right here, this top line, that represents the learning curve of the young adults. The bottom line here represents the learning curve of the aged adults. Now, two things to know. One is that by the end of the session, there was no difference in performance. The young and the aged subjects performed the same. What's unique is that it took the old individuals a little bit longer to learn the task. And we think that reflects the fact that you were using perhaps less efficient but alternate strategies to learn the task, perhaps with the help of those compensatory neural mechanisms I just described in the previous slide. Now, if you look at these last 2 points and plot them right here so that it just shows that the percent correct on the last 2 trials are the same between young and aged subjects, and then now you test them on those flexibility trials. What you find is that when the subjects are shown novel pairs, that's a little harder. And so even the young subjects go down in performance. Okay? But they still do not too badly. They're performing at about 80%. The old subjects, on the other hand, drop much more dramatically, suggesting that they have a little bit less flexible memory systems available to them. So while those neurons can compensate for some of the losses, the cellular losses in an older brain, that they may be challenged when asked to perform flexible kinds of tasks. Okay? So there's many different future directions for memory research, and I've highlighted some of the directions that would be of interest to probably a very broad community. One has to do with the question of enhancing memory. It's going to be a challenge because, as I said, different memory training regimes will not work on everybody. How do we selectively treat memory dysfunctions? Again, there may be different causes of memory disorders. There may be different reasons why you don't remember information. Again, this will be a challenge, but I don't think it's insurmountable. No, it's also different, which you may be less aware of, is that a number of researchers are very interested in the interaction between memory systems and decision-making systems because they're trying to develop autonomous, smart robots, ones that can learn from their behaviors, learn about the consequences of the behavior, update their, quote, memory networks, and then have the new memory networks drive the future behaviors. And then it looks at the consequences of those behaviors. Those go back and update memory to drive the next set of behaviors. As you can imagine, why? The military is very interested in these kinds of robotics, and there's a big push in the military research to incorporate a lot of the spatial learning and as well as the decision-making research that I just talked about into their plans. Thank you.